Welcome to Whatever You're Thinking, Think Smaller. My name's Kim Killian, and I'm the Regional Sales Manager for the Southwest Territory for Precision Nanosystems, celebrating 10 years creating transformative medicine. Today, I'm speaking with Dr. Gaurav Sahay, who is an assistant professor in the College of Pharmacy at Oregon State University. Our focus today will be on lipid nanoparticles, specifically the evolution and usage of the LNPs. Before we dive into lipid nanoparticles, I'm curious what your earliest memory is as it relates to science.
Yeah, I think, for science, I think, I was involved in some science fairs where I remember one time, there was a quiz and the guy who was supposed to be in that quiz basically couldn't make it. And I was in the audience and they put me on there and like out of the fluke, basically we did really well in that quiz. I had that tendency to sort of like look into science, but initially, like in the childhood, I thought I'll become an actor, then a scientist, and then it changed to becoming a director. And I do think science and is sort of like very creative process, like making movies where you have a script, so you write a grant. In our case, you go to a producer like a grant agency, then you hire assistant directors like our grad students and postdocs. Then you work with stars. So I've been working with lipid nanoparticle when he was not that well known. Now he's becoming a superstar, and RNA technologies. And then you do experiments, take pictures, find data, put that in a research publication, and that's basically our movie, and set it out. So, and how you present it, how you go about presenting, how you take those different questions and put into a story is very creative process. And as a scientist, I think that sort of always drove me in terms of like creativity, even looking back.
So if you had to pick your most favorite scientific word, what would it be?
NanoMedicine.
That would be your favorite word?
Yeah, absolutely. I think we, again, we work in this area because, of course, we are passionate about it. And I think we are seeing, such transformation happening, right, in the field of nanotechnology. And I think in later half of the podcast, we might be talking about, lipid nanoparticles and how important sort of they have become in delivery of, mRNA vaccines for COVID. A lot of these technologies are, right now we are in the midst of a real-time experiment, basically, right? So you see in the world, basically how science works, where people are doing different stuff and then that gets reported, and it's like a whole big lab experiment happening where real-time data is being generated and it's just amplified. And I think nanomedicine has a key role now in that, not just vaccines, but detection, and you'll see a whole lot of other arsenal. And we have been there for cancer and for other disorders as well. So I think it's a very topical area, and the full potential, even though we as scientists state that in grants and papers, but you could see this happening in real time. So I think it's a very important time in the field, as well as you can see the importance of it in terms of you can engage students to see what we see as well as the power of nanotechnology in a lot of medicine as well as diagnostics.
Maybe could you talk a little bit more about the evolution or usage of lipid nanoparticles?
Lipid-based nanoparticles have been around for a long time, at least 40, 50 years ago. Genesis started, you can go back as like Alec Bangham, who, started to use liposomes. But let's start with, a little bit from the current towards backtrack the history. So for example, Moderna recently said, within 63 days, they were able to package a mRNA vaccine in a lipid nanoparticle. And I began thinking that, first of all, I think it required a lot of prescience on their part, where, they had a manufacturing system built, and they were working on it, on infectious diseases. But that journey of 63 days took a long time effort. It took a 10-year effort for Moderna to bring the capability from where it is. So if you go back, this would have been a 50 or 20 years effort, because there was a company called Alnylam which started working on these technologies and brought that to clinic. So you had some de-risking done during that process. But if you even go back then, when sRNA was coming into picture, the basic knowledge was derived from technologies that were being developed for DNA. There's some elegant papers where it all started. You have Peter Cullis and Frank Zoka, Li Huang and all those guys working on using liposomes for sRNA delivery, for DNA delivery. Then, Dan Anderson and Bob Langer's like high throughput approach that basically increase the material space. So again, the point I'm trying to make it that this has been a long journey, and the 63-day or 40-day time point, which will all, now what will happen is there will be an arsenal of these technologies coming, and the time point will be 5 days one day. But it has taken a lot of persistence, funding, a lot of great scientists, a lot of students who have brought us to that juncture. And it's a very important thing for us to realize historically of how we have reached to this point.
Dr. Sahay, you're considered a pioneer in overcoming endosomal barriers in intracellular delivery. How does that make you feel to be considered a pioneer in your field?
Yeah, I think, I look to other pioneers. I think, it's great to be given that word, but I think we all in science are pioneers in one way or the other. But I take incredible inspiration from the real pioneers or founders that have come before us. I mean, we'll continue to build on what they had done. But of course, this doesn't mean that research has ended there. I mean, it was just the beginning. So I take inspiration from them, and hopefully we come up with, new ways, go beyond the current sort of thought process that is, maybe something that is on the fringe, and, try to understand that and bring it to life. I think that's just an exciting part of being a scientist.
Maybe could you elaborate for us on some challenges you find delivering APIs into cells?
Of course, for nanotechnology, one challenge is that it has been liver, or, now it is in the immune system specific. But reaching the brain is tough. Reaching certain part of the lung after inhalation is tough. You inject something in the eye, it just stays in the, front of the eye. So we have to come up with new strategies to overcome some of these biological barriers. But I think for intracellular barriers, once things are internalized, most of the material that you see, even with the most potent ones, whatever, is in the clinic right now, the nucleic acid delivery to the cytosol has been very limited. So I think if, again, like we look into, how these different viruses are doing it, I think getting some bioinspiration there might be helpful, but we'll have to come up with a way that at a cellular level, especially when you are dealing with breaking the endosomal membranes, not really breaking, but escaping it, most of the materials, not just nucleic acid, but small molecules are stuck there. But there might be ways, and we might look into nature to tell us because, it has billions of years of experience to find a way to escape that. So I think, and at the intracellular level, that is basically a problem. But for the drug delivery field, I think the problem is delivering materials which are extra, not just in the hepatic region, even locally. One might think eye is easier, but it's still not. So there are a lot of technologies that we need to develop, and fundamentally understand as well.
What are you most curious about right now?
Like everyone else, I think the virus is in everyone's mind right now. I think you'll be seeing a lot of technologies coming in. As I said, like people in nanotechnology, it's a deadly disease, but the structure, my compliments to the chef, that is nature, for revealing these kind of structures, because not just we fight them, but use them in some way that can, help in mitigating certain kind of disorders. So I think basically right now, even though we are, working on what we passionately believe in, and pre-pandemic, but I think in most of our minds, the virus is in like everyone's mind right now, and how we can resolve the problem, how nanotechnology we can impact, what new things that we learn. I think that's certainly where the curiosity is high.
It's been a delight speaking with you. Thank you very much. You made my day. I hope you have a great day.
Thank you so much, Kimberly, and thanks for this invitation. Do keep in touch.
Thanks for joining us. And remember, whatever you're thinking, think smaller. And to learn more about Precision Nanosystems, please go to our website at www.precisionnanosystems.com.